I would like to start my talk with a concept that's a little bit behind the Human Brain Project, because we understand the human brain as a system that is organized on different spatial but also temporal scales, from the molecule, from the genes to small circuits, as you know, to the so-called mesoscopic scale, and to large networks that are involved in a particular cognitive function or in consciousness. And we can obtain from the brain different types of information and study the brain using different empirical models, methods. We can also develop theories, but there is more and more also coming in the second aspect that we can use and develop methods for data analytics that we are based on theory and mathematical models, performing simulations that then are checked again against empirical research. And I show this slide, and perhaps some of you have seen it already, because it shows the ambition. Yeah? So when we want to understand the human brain, it's really from cortical microcircuits to consciousness or even below. So we need to understand the different relationships that the different scales have with each other. And we have very often methods that are targeting only a specific level, and then the question is, how do we connect it? How do we know that when there's a certain receptor change, a certain channel is changing, that then a microcircuit is becoming active, and how many microcircuits we need in order to perform a cognitive task, and how these things are linked together. So that's a very large approach, and this is also, in my view, well reflected in the Human Brain Project, which is a flagship project with a 10 years duration. It was started in 2013 and had this large ambition, yeah, to contribute to a deeper understanding of the human brain, but do it in a specific field that is at the intersection of computing, neurotechnologies, and medicine. And this is something special. So it's not just a neuroscience project, it's really a project that tries to make an impact at this intersection. And of course, since the brain is so big, so complex, we cannot do everything in one and the same time. So we decided in this last phase, which is called SGA3, and it started in 2020, we started to focus on the connectome, but again, taking the connectome in its broadest understanding. So this is the hardware, yeah, this is also activity, these are molecular changes, and we would like to understand the connectome with respect to its intersubject variability, but also the role that the connectome plays when we see a certain cognitive process or when we want to understand consciousness. And last but not least, we identified as a particularly important point that is highly relevant to use it for adaptive architectures of cognitive functions, so also to translate knowledge from neuroscience into deep neuronal networks, for example, or into neuromorphic hardware, or into neurobotics. And this is a new field emerging, and I think many experts that are working in the HBP and many students that got education in this context, they have really nice perspective for future research and for future work. So how do we do it? Our strategy is so-called co-design. And I would say we invented the co-design in the neuroscience. And as co-design, we mean that we start from a neuroscientific question. We develop together with engineers and informaticists tools and softwares that we call now e-brains. And we contribute also high-quality data, but then also test it and help others to use it. And this very close interaction is extremely important, and probably many of you have seen that very nice and expensive infrastructures have been developed in the past and then nobody used it, because they were too far away from the needs of the neuroscience. And here the idea was that from the very beginning they work hand in hand. So eBrains is openly accessible to the international research community, not only limited to Europe, of course. And we are now on the so-called S3 roadmap. That will mean that we are developing towards sustainability so that eBrains is also active and can be accessed when the Human Brain project will be finished in 2023. So at a glance, the HBP, these are 122 partners from 17 institutions. We have about 500 researchers directly involved. There are many partnering projects which are really nice because they bring in research questions from outside the HBP, and there are really exciting projects running. We have published, meanwhile, 2,000 publications, and it's really going up, so it's increasing the speed. We had an estimated funding of 406, so sometimes we read in the newspapers that it is 1.2 billion or even more. In fact, it is 406 from the EC, and it was thought that the remaining amount is contributed by in-kind contributions. So many of examples of the work will be presented in the next days and 2 days. So I will not try to cover everything. That would be simply a task that cannot be addressed. Listen to the talks. I will give you only a very few examples where I think they are exemplary for a certain type of research. And I would like to start with an example from our friend and colleague Javier de Felipe in Spain. He is doing research on the brain's ultrastructure. That is, he is doing intracellular injections of Lucifer yellow, for example, in fixed human cortical tissue, and then he is analyzing it with very powerful optical methods, including electron microscopy. And there's a big progress, I would say, since Javier is now capable to do this in human brain tissue, and this is not self-running, so it's extremely difficult to have such experiments even in mouse and rat, and in human brains with a post-mortem delay it becomes really tricky. But he has now rather fresh tissue available, and he is doing these very nice mappings of the ultrastructure of the human brain. And many of his data are already now accessible in the neuromorph.org organization. This is a very large database that has lots of cellular data, physiological data, where do they come from, what is the dendritic length, what are the branching pattern, and so on, subdivided by different cell types, cell regions, species, and so on and so forth. So that's a US effort where he is contributing for a long time. But these data are also accessible through eBrains, and they are being used, and they are the basis to develop new models of pyramidal cells, for example, and to inform simulation running on eBrains. And 2 examples here are given, 2 recent publications with respect to the structure of the hippocampus of cells in CA1. And Michele Migliore, I'm sure, will tell us more than about it, how his models are being informed by true biological data. And this is prerequisite to make the simulation so powerful and to really reflect features that we can also extract using physiological experiments, for example. And the other example is from stellate cells, computational modeling, where also this type of information is used to predict certain features. And I'm convinced, and so are many colleagues from the HBP, that we have, so to say, to make iterative loops. So we are doing, as neuroscientists, experiments. We develop together with our colleagues models and simulation, and then this should go back, of course, to empirical experiments. And in the best way, this simulation informs the empirical experiments, what could be done, but perhaps also what could be not done, because it simply does not make any sense which can be a result of a simulation. But again, this interaction of simulation modelers, I would say, and empirical neuroscientists, this is something that has been developed in the past years, and that is absolutely necessary that, so to say, we speak with each other. So we are developing these new models and have this type of interaction. But you can also imagine that's not so easy methodically to implement it. And therefore, eBrains is a right research infrastructure and a platform to collaborate. And I showed you already two of its aspects, that is data and knowledge. So we have data like from Javier de Felipe's lab that are being annotated, that are being curated. They have an extremely rich annotation. This takes time. And I should say we underestimated a little bit the time that is needed in order to bring in data into the platform. But it makes a lot of sense, because when you properly annotate your experiments, then others can use it. And if not, it's simply only a storage, and it's not helpful. And simulation, I have also mentioned already, there are also brain-inspired technologies like neuromorphic computing and neurobotics, and I do not have time today to mention examples here, but Walter Senn is here and hopefully will bring us more information. And we have medical data analytics where we support clinical research. So let me focus a little bit on the atlases. And the atlases have the idea that we want to cover the different spatial and temporal scale and provide the atlases as an integrative platform. And the idea or the concept is illustrated here. So we start from cytoarchitecture, from the cellular architecture, because the cells, these are the element that process information. So we need to understand how cells are related to each other, what is their morphology, their physiology, but also what is their architecture, their pattern in the brain. And we see that the cerebral cortex and subcortical nuclei, they have a very specific distribution and arrangement of cells. We call this architecture. And then we do such type of analysis not only in one brain but in 10 brains, because brains are different. There's intersubject variability, and in the same way as we differ between each other with respect to our height and eye color and hairs and so on, brains have the same way of, or the same magnitude of variation, and this is of course extremely relevant, at least when you think about patients. Yeah? You want to be precise and make predictions for a certain patient and not for the average patient, so that would not work. Then we would like to bring together the cytoarchitecture with a function, for example, as we can obtain it in functional magnetic resonance imaging, and Lars Mukli is here, and he is doing very sophisticated experiments for the visual world, for example. Then we have a third aspect, that is receptors. So receptors are key molecules for signal transduction. Receptors are the places where a certain transmitter is binding, and there are excitatory receptors, inhibitory, modulatory receptors. They act at the cellular level, and this is important to understand what the processes are, and Matthew Larcombe will show more about this on the at the cellular level. But it's also important when you think about diseases, because most of the drugs are acting to receptors, and we should understand their distribution. And last but not least, there's another aspect, the transcriptome. So when we want to understand causal mechanisms and want to understand why something happens in the brain, we need to go to the genes down. And I could, of course, enlarge the series of aspects, but let me focus on these four at this point. So we start with cytoarchitecture, and this is an image from the superior temporal gyrus, more precisely from the Heschel gyrus, where the primary auditory cortex is located, and from the middle frontal gyrus, where higher language areas are located and the presumed Wernicke's region is located. And you see here a cross-section in a cell body stain section with higher magnification. And without going into detail, you get probably a flavor that the architecture of cell is different in the pictures left and right. The laminar pattern is a little bit different. The columnar arrangement is a little bit different. And we use such parameters in order to describe the cytoarchitecture in quantitative terms. So we measure such cytoarchitectonic differences and are then able to identify borders between cytoarchitectonic areas, which are labeled here by different colors. And I would like to focus your attention, or to put your attention to the fact that the borders of the areas do not coincide necessarily with the sulci. So some borders are located in the depths of a sulcus, but usually they don't. Usually they are somewhere. And that means that the sulcal pattern and the gyral pattern is not a good predictor of the localization of an area. But when you move to another area, you move to another connectivity, you move to another receptor architecture, you move to another function. So it's important to understand where precisely the areas are located. And sulci are no good predictors for, I would say, 90–95% of the areas in the human brain. So we map such areas in 10 human brains in serial section, reconstruct the sections, and then compute so-called probabilistic maps, which quantify for each position in a certain reference brain the probability with which a certain area is present. And here in the upper slides, you can see probability maps of area STS2, because it's located mainly in the superior temporal sulcus, and 2 because there are more of them. And red means there is a high probability that this area is present there, but blue and green means there is only 30 or 10 or 20% probability that you find this area in this particular position. So they are located here, and then we compared it with the result of a published study that was a diffusion imaging study that postulated an area SMTG based on connectivity patterns, and it coincides in size and localization very nicely with STS2. So this shows you that there is indeed a very close link between connectivity and cytoarchitecture, and the maps can be used to verify certain findings from imaging, but it can be also used to have a certain experiment and to search specifically for an area that has not been identified. So in the same way, we can compare the cytoarchitectonically identified areas, which are indeed listed here on the right side, with functional imaging data, and this is a meta-analysis, and what we did was we have different paradigm classes and associated it with certain areas in the temporal insular region, in the primary auditory cortex, in higher auditory areas, and in the superior temporal sulcus areas. And just looking to the pattern, you can see that the insular areas, so those which are between the hesial gyrus and the insula, they have a quite different functional pattern as compared to the primary auditory cortex, and I would say also higher auditory areas. But when you go to the STS areas, the pattern changes. And you see that higher functions, including even theory of mind, episodic recall, reading, and so on, are being involved. So that means there's also a functional difference. And of course it has some limitations when you think about spatial resolution of functional imaging studies and meta-analysis. But when you have very good functional experiments, then of course you gain a very high accuracy of your research, and then you can go more and more into finer grained parcellations of the human brain. So we can also go downstairs the spatial scales, I would say, and in this study that is now in press, we analyzed the gene expression of functional systems. 4 functional systems where we addressed the motor system, somatosensory, visual, and auditory cortex. And we have analyzed in each functional systems several areas. From the primary area, this is area 17 or OC1, going to higher visual areas. Or in the auditory cortex, the same, from the primary auditory cortex to higher auditory and associative areas, and the same also for motor, premotor cortex, and so on and so forth. So we used the cytoarchitectonic probabilistic maps as seed regions, as regions of interest, and then linked the maps to the Allen Brain Institute data and selected from this atlas gene expression data. So there is a tool provided by eBrains that allows you to do it. And we selected those genes that are involved in the expression of certain receptor types in the brain. And then in the second line of study, we studied receptor autoradiograms where we quantified the concentration and the density of certain receptor types responsible for binding certain neurotransmitters. And this was a large series. We studied about 15 to 20 different neurotransmitter systems. And our idea was, what is the relationship between the gene expression and the receptors? And we did not, or my lab did not analyze gene expression. So I was happy to rely on this resource from the Allen Brain Institute and to study this relationship. So it's a combined study. And the result is shown here for only a few examples. So these are the areas grouped according to gene expression. So we did a cluster analysis based on a lot of genes coding for certain receptor types in different functional systems. And you see that the visual systems form a cluster, the auditory systems form a cluster, the somatosensory systems are here and the motor are there. And interestingly, the clusters that we can find based on receptor densities, completely different data set, completely different type of data, it reveals the same pattern. And we can even identify gradients within a system coming from the primary visual areas to higher visual areas like the force in the dorsal stream area. And the same is not so clearly true, but the same is here also seen for the auditory cortex. And at least in these functional systems, we see a high degree of covariation between receptor types and receptors. And this is interesting because several receptors are coded by many genes, yeah? And also those have a certain covariation, at least in these functional systems. When it comes to associative areas, this might be a bit different and it might be a weaker coloration, but for the higher, for the primary sensory areas and motor areas, this is a case. And to enable this, we have developed Jülich, which is a tool that allows you to connect cytoarchitectonic data with gene expression data, and it talks to Seattle, so to say, and extracts the receptors in those regions that you identify. And there's a very nice and intuitive approach to do it by click and to do the analysis online, but there's also a Python-based possibility, so you can also have a more programmatic express and can write your own Python scripts in order to do such type of analysis. So the Atlas of the Human Brain is developed in order to study the relationship between structure and function or dysfunction, and this is, I think, very much in the focus of this conference here. And there are different tools that are available, like this viewer or like a new surface-based map of the different cytoarchitectonic areas, and we are working on linking this map now to free surface. But for every single area, we have additional data. Not every area has all data, but there's a lot of data. For example, connectivity profiles or receptor densities for many neurotransmitter receptors or cellular densities. So that's a way of an atlas that, in my view, it's quite new, because it's not only a where atlas, it's also a what atlas. So you can also learn about the different brain region and inform your own models and theory. And this is done, for example, in Victor Gilza's work, and he's the next speaker, so he will probably also comment on this. But the idea is really to bring different words together and to get a new quality of research, I would say, by informing us. And no lab, no group is capable enough to study everything and to do genetics and receptors. So it makes a lot of sense to join forces and to use this infrastructure. At the very end, I would like to put your attention to a paper that is discussing the upcoming decade of digital brain research. And this paper has been initiated by the Science and Infrastructure Board of the Human Brain Project, and we worked in the SIB for quite a long time. We had very heavy discussions, subgroups, and hot debates, I would say. And then we shared it with the work packages. But the idea was from the very beginning to include as many of you as possible. This is an open paper. This is a dynamic paper. So we would like to invite you to visit the HBP website and join the discussion. And if you feel something is missing, or if you feel, well, that is not so nicely represented as you would think about, or you would say, no, you have to write it different, then please let us know, and we will have a discussion, and then we, so to say, you can also be an author of this or a co-author of this paper. So we will have regular updates of the papers with new authors. And if you think, well, I do not like this paper anymore, you can also be removed as an author. So it's a completely, I would say, community-driven effort where you can help to shape the future of digital neuroscience. And I think this is absolutely necessary also to be noisy and to articulate what we want as researchers, and many of you are working with digital tools. Because we want that our research is also funded, that our ministries and our funding organizations also are aware that this is an important type of research. Yeah? So please go there, see what can be done. If you just like it, become a supporter. This is also possible. And I would really hope that this paper is further developed, and in the next Human Brain Summit, which is a completely open summit in Marseille next year, last week in March, we meet in Marseille. We will have a final discussion, and I really hope that we will have an impact with this paper. So please join us, doctoral student, postdoc, professors, and help us to make this paper really a major reference. And saying that, I would like to thank you for your attention and for being here today.